So, student, welcome. In last lecture, we have learned about nucleotide. Now, today, we will learn about the replication of DNA. Before learning or before studying replication, we have to recall DNA. In last year, you have seen DNA. DNA is a part of nucleic acid that is present in a nucleus, and DNA is a genetic material. We have proved that by seeing the experiment which have performed by Hershey and Chase, by Griffith, by McLeod, McCarthy. So this DNA, deoxyribose nucleic acid, has, if you see the structure of that DNA, it is a double-stranded helical model, in which two strands are there, which are made up of nucleotides, and these two strands are coiling on each other. They are coiling each other, and they are attached by the nitrogen bases. They are attached by the nitrogen bases of each nucleotide and of each complementary nucleotide present on separate strands. And between these two nitrogen bases, there is a hydrogen bond, and due to this bonding, the strands are attached to each other and they form a ladder-like structure. So, such is the structure of DNA, in which the DNA strands are made up of nucleotides. And if you see, one nucleotide is made up of three constituents, that is pentose sugar, in DNA it is deoxyribose sugar, then phosphate group, and third nitrogen bases. Nitrogen bases are of two types, purines and pyrimidines. In purines, we have seen that it has again two types, adenine and guanine. And in pyrimidine, there are two types, that is cytosine and thymine. So, these nucleotides, one nucleotide is made up of three constituents, and these nucleotides get attached to one another by phosphodiester bond. Which bond is present? Phosphodiester bond is present between each nucleotide, which attach it and it forms a strand. Secondly, the bond between sugar and nitrogen base, that bond is called as what? Glycosidic bond. That is called as what? Glycosidic bond. And bond between nitrogen bases, complementary nitrogen bases. Suppose adenine, it make bond or it pairs with thymine. There is double hydrogen bond between them. And cytosine and guanine, they pairs together. They are complementary to each other, and there is a triple hydrogen bond between them. So we have seen this in last year in 11th standard. So this is a recalling of DNA. Now we will see DNA replication. Now if you see, in last year we have seen mitosis. In mitosis, if you see, it is an equational division in which from one cell, two cells are formed. And the chromosome number of this parent cell and this daughter cell is same. Because of what? Because when this cell has to divide, it goes under the interphase. In interphase, before division, there is a preparatory phase, and in this preparatory phase, the DNA duplicates cell organelle get duplicates. So has that there should be a equal material, equal number of DNA in this, equal number of DNA in this means equal number of chromosomes in this, equal number of chromosomes in this means has so as of parent cell. And in cell cycle, if you see in cell cycle, there is an interphase and one division phase is there. In interphase we have seen that in cell cycle, in interphase we have seen that there is a G1 phase, S1 phase and G2 phase. Growth phase number 1, this is the synthesis phase and this is the growth phase number 2. In this S phase, there is a replication of DNA. And in one DNA, there is only one time replication, or in one cell only there is one time replication of that cell. And it happens in what? S phase. And S phase comes under interphase. And interphase comes under cell cycle. That is the preparatory phase. Preparatory phase. Preparation for division. In preparatory phase, what is there? There is a preparation so that the material which is present in the parent cell should be equal in the daughter cell. So this is so you should keep in your mind that replication occurs in the S phase of interphase and it occurs only once. It occurs only once in that particular DNA. Only particular DNA. So now we will see DNA replication. Now if we say that DNA regulates and controls all the activities in the cell. When cell reproduces, cell should duplicate, and this cell duplicates due to the replication. This cell duplicates due to the replication. Has a carrier of genetic information, the DNA has to perform two important functions. As a carrier of DNA genetic information, as a carrier of genetic information, as a carrier of genetic information, DNA performs two types of functions. First is heterocatalytic function. And second is autocatalytic function. Now if you see heterocatalytic function, when the DNA directs the synthesis of chemical molecules other than itself, when DNA directs the synthesis of chemical molecules other than itself. Example, synthesis of protein or other molecules, that is called as what? That function is called as what? Heterocatalytic function. And what is autolytic? In which it directs, DNA when it directs the synthesis of chemical molecules of itself only. Direct the functions of itself, then it is called as what? Autolytic function. Example, replication. The function which is directed by DNA when it duplicates itself, then that is called as what? Autocatalytic function. So this is two types of function which is performed by DNA. Now we will see the replication process. Now what is replicon or what is replication? Suppose if you stand before the mirror, what you will see? You will see your mirror copy. That mirror copy is called as what? Replicon or replica of yours. So that is that when a DNA synthesizes the molecule of itself, mirror copy of itself, that is called as what? Replication. And if we see this replication occurs in a prokaryotic cell also and eukaryotic cell also. In prokaryotic cell, that replication is called as what? Theta replication. And if you see in prokaryotic cell, there is only one circular molecule of DNA and there is only one nick. Only at one point there is a cut given to this prokaryotic cell. We will not discuss about that because we have the point of eukaryotic. We will see in detail the eukaryotic replication. Eukaryotic DNA replication. Now, if we see in a eukaryotic cell, the DNA replication is semi-conservative type of replication. And how it is semi-conservative, I will tell you at the last. When we will see full process. The 7 steps we have to see. Now, in DNA replication, first step is activation of nucleotide. Now, I have told you what are nucleotides. Nucleotides are the units of DNA, or the DNA is made up of several nucleotides. So activation of nucleotide. And if you see, in last year I have explained you or you have learned that the name of nucleotide is given by the nitrogen base which is present in that particular nucleotide. And according to the presence of nitrogen base, there are 4 types of nucleotides. That is adenine nucleotide, then guanine nucleotide, then cytosine nucleotide, then thymine nucleotide. So, before replication, these nucleotides are in deactivated stage, and for division or for replication, they have to be activated. Which in a deactivated form, they are present in which form? AMP CMP and TMP. TMP. Adenine monophosphate, guanine monophosphate, and deoxyribose. D stand for deoxyribose nucleotide. D stand for what? Deoxyribose nucleotide. So adenine monophosphate, guanine monophosphate, cytosine monophosphate, and thymine monophosphate. This is the deactivated state. Now these are activated by ATP. Means phosphorylation is there. Means phosphorylation is there. With the help of which enzyme? Phosphorylase enzyme. And conversion of AMP is converted into ATP, then GTP, then CTP, then TPP. TTP. Adenine triphosphate, guanine triphosphate, cytosine triphosphate, and thymine triphosphate. This is what? Activated stage. This is activated stage, and this is called as what? Phosphorylation. This is called as what? Phosphorylation. And which enzyme helps this process? Phosphorylase enzyme. Which enzymes help this process? Phosphorylase. So in first step, what is there? There is activation of nucleotide with the help of ATP. ATP is the energy molecule, or it is the currency of energy. And with the help of ATP, this deactivated nucleotide has to be activated, and the activation is known as what? Phosphorylation, and this occurs due to the action of phosphorylase enzyme. So this is the first step. Now we will see the second stage. Origin or initiation point. Origin or initiation point. Point of origin and initiation point. Second step. Point of origin or initiation point. If you see in prokaryotes, in theta replication, there is only at one point the DNA is cut. DNA can be cut. But if you see the DNA in eukaryote, it is long and long. And there are one certain points or certain sites on that DNA. That sites are called as points of origin or initiation point, on which a nick is given. And due to this nick, there is cutting of that DNA with the help of what? Restriction endonuclease enzyme. With the help of what? Endonuclease enzyme. So in this point, if we see the specific point O is present on that nucleus. Suppose this is a DNA. Particular sites are there on this, and at this particular site, what happens? This extends, and at certain point, this point is called as what? O. This is origin. At this point, endonuclease enzyme gives cut, and that endonuclease enzyme is called as what? Natural scissors. Is called as what? natural scissors. This is the O point. At O point there is a nick given. By whom? Nick. By whom? Endonuclease enzyme, and with the help of endonuclease enzyme, this DNA is broken. This DNA is broken. And these two strands get separated. These two strands get separated. So, if you see point of, this is called as what? Point of origin. And with the help of it, endonuclease enzyme, a nick is given and sugar phosphate bond breaks. Sugar phosphate bond phosphate bond, or that is called as what? Phosphodiester bond. Phosphodiester bond, or it is also called as backbone of that nucleotide or DNA. Phosphodiester bond breaks and strand get separated. So this is the point of origin, the point at which the nick is given by endonuclease enzyme. Such points are present on the whole molecule of DNA at certain point. This specific site is called as what? Restriction site, because with the help of a restriction endonuclease enzyme, a nick is given and DNA is cut there at that particular site, that is called as a restriction site. So this is the second stage. Now we will see the third stage. Unwinding of DNA strands. So after giving the nick by endonuclease enzyme, the DNA strands unwind. They starts to separate each other, and this is done by whom? Helicase enzyme. This unwinding is catalyzed by whom? Helicase enzyme. Due to the helicase enzyme, unwinding is there. DNA gets separate and it forms what? Y-shaped structure, that is called as replicating form or replication form. So helicase enzyme brings about the unwinding of DNA, which is cut at certain sites, and this unwinding is done by helicase enzyme. Now if you see this DNA, when unwinding is there, they tends to rewind. But this rewinding is not allowed by SSBP, or rewinding is prevented. Rewinding is prevented by SSBP. What is SSBP? It is a specific protein, and that protein is called as what? Single-stranded binding protein. Single-stranded binding protein. Next, if we see this DNA, the unwinding is there, and due to this unwinding, a Y-shaped structure is formed, and this unwinding is prevented by SSBP. This unwinding is prevented by SSBP, and this unwinding is controlled by whom? Helicase enzyme. This unwinding is controlled by whom? Helicase enzyme. Now we will see the fourth point, that is replication form. replication fork. This Y-shaped structure which is formed, this Y-shaped structure which is formed is called as what? Replicating fork. Replication fork. Replication fork. And both this template, both this parent DNA, separated parent DNA strands are known as what? Template. They are known as what? Template. And I will give you a simple example. When we stand before a mirror, mirror shows our coffee, and mirror, we are the template there. We are the template there. Such is the condition of this. Because due to this template, a new strand has to be synthesized. New strand synthesized, which is mirror copy of the parent strand. And so this parent strand is called as what? Template. This is called as what? Template. So this is the fourth step in which a replication fork is formed, and in this state, this unwinding and the replication fork have a strain, little strain, and this strain is relieved by superhelix relaxing enzyme. Strain is relieved by superhelix relaxing enzyme. Superhelix relaxing enzyme. With the help of this enzyme, the strain which is created in this DNA molecule, or due to the replication fork, there is little strain, there is a pain there, and this pain is relieved by superhelix relaxing enzyme. Superhelix relaxing enzyme relieve this strain. So this is the fourth step. Now we will see the fifth main step. Fourth step is synthesis of new strand. Main point in this replication, synthesis of new strand. Now what happened? I will draw this figure here now. I will mention all the points in this figure. If suppose this is 5 dash, it is also called as 5 dash or 5 primer, 3 primer, or we will call it as a dash. 5 dash, this is 3 dash, this is 5 dash. Because we have seen the DNA strands are anti-parallel and complementary to each other. Means if suppose one strand run in this direction, second will run opposite to that of the first. So we will see the synthesis of new strand now. What happens in the synthesis? Now we have seen that here on this template strand there is presence of what? Nitrogen bases. If suppose A is there, here T is there, here C is there, here A is there, here T is there, here G is there. So these are the nucleotides here. Nitrogen bases are there. Now what happens in this state? A RNA primer get attached here. RNA molecule, or that is called as what? RNA primer. RNA primer get attached to this, attached to the 3' chain. Now, what is there after attaching of RNA primer? Due to this effect of RNA primer, with the help of DNA polymerase enzyme, from the surrounding nucleotide or from the surrounding nucleoplasm. So this DNA is present in what? Nucleus. This DNA is present in what? Nucleus. This is a nucleus here. Around this nucleus there is a cell here. So the material inside this nucleus we will call it as nucleoplasm. So what happens? This RNA primer, what does it do? It collects or it synthesizes the complementary nucleotides from the surrounding nucleoplasm. If suppose it synthesizes thymine nucleotide, the thymine nucleotide will make bond with this adenine nucleotide. If it collects adenine nucleotide, it will make bond with this. If guanine nucleotide is collected, it will attach here. If thymine nucleotide is collected, it will attach here. If adenine nucleotide is synthesized, it will attach here. If cytosine nucleotide is collected, it will attach here to the complementary nitrogen base or to the complementary nucleotide, and strand is formed. And always you kept in your mind that the new strand which is formed in the DNA or is found in replication, this is in which direction? 5' to 3'. 5 direction is 5' to 3'. It is directed here, 3'. So a new strand is formed. But at the opposite end, what happens? There is not attachment of only RNA primer, one RNA primer. There are several RNA primers get attached, and the strand which is formed is strand here one primer get attached, here one primer get attached, one primer get attached, because RNA primer attaches to 3' end. Where will here 3' end is present here. So this 3' end directs the synthesis here, and a fragmented form of DNA is formed. Fragmented form of DNA. These fragments are called as what? Okazaki fragments. Okazaki fragments. And the name given to this fragment is after the scientist Okazaki. So these are the Okazaki fragments. But after completion of this process, what happens? This RNA primer is removed, and this discontinuous strand or these fragments are joined by whom? Ligase enzyme. These fragments are joined by ligase enzyme, and this discontinuous strand is make continuous or a full strand is formed. And these fragments are also complementary to the nitrogen bases or to the nucleotides which are present here. According to them, they are synthesized. So the end at which a continuous strand is formed, or the 3' end at which the RNA primer attached, that there is a continuous strand formed, and so the process begins here. So this strand is a leader here. It gives lead to the formation of DNA, new DNA strand. So this is called as a leading template or leading strand. And this is called as what? Lagging strand. This is called as lagging strand. This is also called as what? Discontinuous strand. Discontinuous. And this is called as what? Continuous strand. This is called as what? Continuous strand. So, the synthesis of new strand occurs due to the RNA primer which attaches to the 3' end and it attracts complementary nucleotides from the surrounding nucleoplasm, and this reaction, this synthesis is catalyzed by whom? DNA polymerase. And which polymerase? DNA polymerase third. DNA polymerase enzyme help the process of synthesis. And main RNA primer. This RNA primer synthesize nucleotide, complementary nucleotide from the surrounding nucleoplasm with the help of DNA polymerase, and there is a synthesis of new strand. And the new strand which is formed is always in 5 to 3' end. 5 to 3' end. This is 5 to 3' end, and this is 3' end, this is 5' end. Now, leading and lagging strand, 4th step, I have explained in the figure. 4th step is lagging strand and leading strand. Two strands are formed. One is continuous, that is called as leading strand, which is formed on the template of 3' end where RNA primer get attached, that is called as leading strand. And the lagging strand, which is formed laterward, and the form new DNA fragments, new DNA which is formed it is in fragmented form, and that fragments are called as what? Okazaki fragment, and that strand is called as what? Lagging strand. That strand is called as what? Lagging strand. So 3' end, at 3' end it is a leading strand which is continuous, and at 5' end that is a lagging strand. They are Okazaki fragments, and these Okazaki fragments later on they are ligated by ligase enzyme, and RNA primer is removed by DNA polymerase first. RNA primer is removed by DNA polymerase first. DNA polymerase first. Because this RNA primer has to be removed to make a continuous strand. So this RNA primer is removed by DNA polymerase first. Now, finally, if you see the DNA guidase or topomerase enzymes form double helix to form daughter strand, daughter molecules. So, the topomerase enzyme or guidase enzyme or topomerase enzyme finally topoisomerase. Topoisomerase enzyme or guidase enzyme finally forms strand, new strand, daughter strand. Form new daughter strand or double helix merge. Then last step is that formation of daughter DNA molecule. Formation of daughter DNA molecule. daughter DNA molecule. So these two strands which are formed, this strand, this DNA separates from here, and after separating, this daughter DNA here in this form, it is revived, a double helical new DNA is formed here. New DNA is formed from this strand, and in these two strands, if we see, one strand is old one, parental strand, and second strand which is formed is a new strand. Means one parental strand is present, one new strand is present. Means half the DNA is made up of parent DNA and one is the new DNA. So a parent DNA is conserved in a daughter DNA. So this DNA replication is called as what? Semi-conservative type of replication. Semi-conservative type of replication. And this semi-conservative type of replication is present in eukaryotic cells. Is present in eukaryotic cells. A replication in which a parent DNA strand is conserved in the daughter DNA strand. And a new strand is present, one parent strand is present, and such a DNA strand, and such a replication in which a new DNA is formed, in which in that new DNA or daughter DNA, one strand is of parent strand and one newly synthesized strand is present, and such replication process is called as what? Semi-conservative type of replication. So we have seen the replication here. In next period, we will see how Messelson and Stark experimentally proved that there is a semi-conservative type of replication. Thanks.